Let's talk about white blood cells now. So another name for white blood cells is leukocytes. Leuko means white in Latin and cyte means cells. Just like every other blood cell, the white blood cells are produced in the bone marrow through the process of hematopoiesis. And the white blood cells are going to be found in tissue fluid as well as some organs. You might remember the white cells are important for immune defense. So some organs are very vulnerable in terms of being exposed to pathogens, like the lungs, for example. You keep on breathing the air in, there could be a lot of pathogens in the air. And so, you want to have some white cells that are stationed there at all times. The white cells can engage in tissue maintenance as well as repairing. And in addition to defending against pathogens, the white cells can destroy dead or dying body cells, right? You might remember just 2 slides ago, I think, the red cells, when they are too old, they will get trapped in the liver and the spleen. And in there, they will be destroyed by something called the macrophage. So the macrophage is a type of white cells, and they will destroy these dead red cells. But it's not just red cells that they deal with. Okay, there are other bodily tissues that are becoming too old or dying. They could get rid of those as well. Now, red cells, they are confined to blood. That means they stay within blood vessels. The only time they come out is if you have a cut in the blood vessels. However, for white cells, it's quite normal for them to kind of squeeze themselves out through the pores in the capillaries. Remember, the capillaries are kind of leaky, right? So the white cells are able to squeeze through there, and they're going to go into the tissue and see if there is any kind of cells that might be infected and that needs to be taken care of, or if there is nothing wrong with the tissue, and sometimes they just go back into circulation. So there are two groups of white cells, and under the microscope, they do look a little bit different. They are very different from the red cells. First of all, right? You can see the red cells are in the background, and the white cells are much bigger than the red cells, and also they have nucleus, and a lot of times they have more than one nuclei actually. But we separate them into two categories. Okay, there are something called granulocytes, and then there are something called agranulocytes, and I'm going to explain that on a note with you. So white cells, we have what's called granulocytes, and we have what's called agranulocytes. So what are granules, right? You might remember from last semester, you learned about cellular structures. So there's your cell, there's your nucleus, and then you might have some of these little vesicles with chemicals in them, right? Those are called lysozyme, right? You might remember lysosome, right? So they digest, destroy things that the cells ingest. So granules are kind of like that, very similar concept in that there are chemicals in these granules, and sometimes the granules are used to, the chemicals are used to break down various things that the cell might have ingested. So the granules are more visible in some white cells than others. So if you stain the slide and you look at it under the microscope and you can see the granules clearly, then we call these cells the granulocytes. Granulocytes. Okay, so they are the ones that are going to have visible granules. Okay, and on the other hand, you are going to have the agranulocytes. Agranulocytes. So they also have granules, but you can't really see them under a microscope. Okay, so granules are not visible. It doesn't mean they're not there, but you just can't really see them clearly. So those are the two kinds of white blood cells, granulocytes and agranulocytes. Let's look at some examples of granulocytes. The first one we're going to look at is something called neutrophils. Neutrophils are the most abundant type of white blood cells you have in the body. Approximately 70% of your white cells are neutrophils, and they are an example of phagocytes. Phagocytes are basically cells that can eat up things, and in this case, the neutrophils will eat up anything that doesn't belong to the body, cellular debris, pathogens in some cases, and they're able to devour them and destroy them. The second one we are going to look at is something called eosinophil. Eosinophil. And this is a type of white cells that is going to attack parasites. So if you have a parasitic worm or something, that would be too big for neutrophils to eat up, and so the eosinophil, they are going to secrete the contents from their granules to attack those parasites. And lastly, we have something called basophil. So basophil, they can secrete something called histamine, which we'll look at in more details later. But basically, that will cause you to have an inflammatory response. So, you know, if you cut your finger, for example, it's going to be painful, it's going to be red, it's going to feel a little bit of heat and maybe a little bit of swelling. All that are basically characteristics of inflammation. And so basophil is a type of cell that is responsible for creating the inflammatory response. So these are some examples of granulocytes. And again, granulocytes are white cells that have visible granules under the microscope. On the other hand, we have the agranulocytes. We have something called monocytes. So the monocytes are basically almost like an inactive type of white cells that are just going to be moving around in the body, and they're going to be surveying the environment. And if they find something that is not supposed to be there, then they will transform into macrophage. Right? And the macrophage will then be able to eat them up. So both macrophage and neutrophils are examples of phagocytes. They can eat things up. Sometimes you would just have a monocyte transforming into phagophage, and they would stay in a particular organ, like the lungs we talked about, right? It's very exposed to potential pathogens in the lungs, so it makes sense to keep some macrophage there. So this kind of transformation is a one-way transformation. There's no going back. Now, once they turn into a macrophage, they will fight until they die. And there are something called the T cells, as well as something called B cells. Right? These we will talk about them in a lot of more details in lecture 3. But basically, for now, T cells, they are responsible for coordinating immune response. And for the B cells, they are responsible for producing antibodies. Right? You might remember antibodies from earlier in the lecture. These are proteins, plasma proteins that helps you fight off infection. So there you have it. 3 examples of granulocytes and 3 examples of agranulocytes, and they are all basically different examples of white blood cells. Let's go back to the lecture notes, the PowerPoints. The next formed elements that we're going to talk about is going to be platelets. Another name for platelets is thrombocytes. So platelets is a little bit different because they are not really a cell, considered as a cell themselves, but rather they are cell fragments that are broken off from a large cell called the megakaryocyte. So here in this picture you see the megakaryocytes, and all these little pink dots, they are basically the platelets that are shed off from the megakaryocyte. So you produce platelets at an astounding rate of 200 billion per day, and they will be able to stay in your body for about 10 days, assuming you do not use them in hemostasis. So hemostasis is basically to stop to stop bleeding. So if you don't have a cut or any kind of bleeding wound for 10 days, then the platelets are going to die. But that's okay. You're going to be making 200 billions per day, so it doesn't really matter. The platelets themselves don't have nucleus, so if we look at all our formed elements, the red cells, the white cells, and the platelets, both the RBC and the platelets, they lack nucleus. Okay, the white cells are the only blood cells that have the nucleus. So this is what coagulation looks like. You're going to form these fibers, and the fibers are going to trap the red cells, and it basically forms the scab that you see, once the bleeding stops. So let's take a closer look at this coagulation pathway. Let's go to our note here, and we're going to look at the coagulation pathway. So coagulation pathway. Coagulation means stopping the bleeding, right? Another word for it was hemostasis, right? Hemostasis. So don't confuse hemostasis with homeostasis, right? Homeostasis is to maintain internal balance, like you shiver when you're cold. Hemostasis is to make sure that the bleeding stops, right? And to achieve that, we need the coagulation pathway. So let's take a look at a blood vessel here. Right, so here is your blood vessel, and normally the blood cells would be passing through, happily, no problem there. And what happens is, if you have some kind of injury, right, like a thumbtack that goes into your blood vessels, and what that does is it's going to expose the underlying tissue, right? And when the tissues are damaged, they release something called thromboplastin. Okay, so this is normally not in your blood. It's only there if you damage tissue. So I'm just gonna put a note here. It's released by damaged tissue. Okay, so circulating in your blood plasma, you might remember from earlier slides that there are two coagulation proteins that are just kind of circulating, and one of them is what we call prothrombin. Okay, prothrombin. Now, if you see the prefix pro in a protein, a lot of times it means that this is inactive. So inactive. Right? It's not really working. Right? And we normally want it to be not working. We don't want it to be switched off unless we have a cut. And to turn this on, we will need thromboplastin. Right? So no cut, no thromboplastin. We're not going to turn on prothrombin. With a cut, with a damaged vessel, you will now have thromboplastin in your blood plasma, and that will help convert. It will help convert the prothrombin into its active form, right? And the active form of prothrombin is just thrombin. So this is going to be active now, right? So green for go, red for stop. So inactive prothrombin will become active thrombin in the presence of thromboplastin. And you're going to need a little bit of calcium for that. You're just going to have to take my word for it. And then another coagulation protein that we talked about was something called fibrinogen. Right? And if you see a suffix of ogen, it usually means it is inactive. Right? So fibrinogen is inactive, and we need to turn it on to stop the bleeding if we have a cut. And to turn it on, we will need to have thrombin there. Okay, so the thrombin, once is activated by thromboplastin, will in turn activate the fibrinogen into fibrin. So this is kind of like a domino effect, right? One activation of one protein will lead to the activation of another protein. And just like before, we're going to need a little bit of calcium as well. So fibrin here is basically the white strand that we saw in this picture, right? All these white network is basically the fibrin strands sticking together, and once they form a web, they're going to be able to trap more platelets. They're going to be able to trap more red cells, and eventually you're going to have enough cells stuck to the fibrin network to seal the leak, and that's how you stop the bleeding. So just as a summary, here are everything we've talked about so far. The plasma portion of blood, what's in it, and their functions. Obviously, you don't have to memorize the percentage. The only percent I want you to remember is that the plasma portion, it makes up 55%, and the cellular portion makes up 45% of your total blood volume. But the actual numbers you don't really have to know. Here are the different types of white cells and their functions. So this is a good summary slide for you to look at.